right, thank you, Dennis. Thank you for the introductions. I'm very excited to present to you today my webinar on optimizing peptide cost of goods with Pure Coli. I'm Erik Nordwald. I lead our process development team on the microbial side. And without further ado, we'll get into it. I'll pause briefly on this slide to give you a chance to take out your phones and scan the QR code for this 2-minute survey, which will help us better serve you and understand your needs and give you an opportunity to reach out to us and connect with us to learn more if there's something that piques your interest or if you'd like to work with us. Today, we'll start with an introduction on KBI and our mission, talk about PUREplatform, set the stage with synthetic versus microbial peptide expression, and then get into some of the data that we have around expressing Liraglutide specifically with Pure Coli, and talk at a more high level about our approach, generally speaking, to expressing peptides, as we understand the landscape is rapidly changing and there's new novel peptides every day that people are interested in expressing, and we believe we can generically deliver low cost of goods for peptides. As far as our mission, KBI is dedicated to building lasting partnerships and delivering innovative solutions to address the most challenging diseases. We ensure quality, transparency, collaboration, and commitment to our partners in supporting the unmet medical needs of patients worldwide. And certainly many of these can be met through peptide production. If you have a peptide you're interested in producing, we'd love to learn more about how we can carry you through the various phases of toxicology, clinical trials, and to market to address various indications that you're working on. As a global CDMO, we have competencies in not only microbial expression for peptides, for proteins, small to large recombinant proteins, but also mammalian expression systems. With over 30 years experience in this area, we were founded in the analytical space. If there's an analytical technique, we most likely have both the capability and the expertise to execute on it. We have over 1,400 different client partners that we've worked with and over 300 drug candidates that we've developed. We're located across the globe with our headquarters in North Carolina, where we have mammalian expression systems, expertise in mammalian expression systems, as well as analytical and process development in this space. Microbially, we're located in Colorado. That's where I am. We have our Microbial Center of Excellence for process development and manufacturing, with scales at 300 liter and 2,000 liter fermentation. That's single use and stainless steel, respectively. Also in Colorado, about a stone's throw away from the Boulder site, is our Louisville site, specifically devoted to analytical. We have a full range of analytics in Boulder to support manufacturing, but we also have supplemental analytics in Louisville and can do standalone analytical work there as well. Finally, over in Europe and Switzerland, we have our Geneva site where we do mammalian cell line development powered by the Solexis technology. Now getting into the heart of the presentation and talking about PUREplatform, which comprises Pure Coli, the main engine behind it all, an overview of the different elements of PUREplatform, and a summary of the benefits, which is KBI's differentiator in this space. In the next section after this, we'll talk about peptide expression and how the PUREplatform really drives down COGS for us and sets us apart from our competitors. Pure Coli has been holistically developed for the manufacture of therapeutic proteins. We took a common grass strain of E. coli and recursively and extensively edited it. About 1,000 cumulative gene alterations we made to the genome to upregulate, downregulate, remove or alter genes in order to optimize the bug, with the result being it grows faster into higher cell densities, which saves you time in plant and is important for cost of goods, as well as the higher biomass inherently gets you off on a higher footing for expressing at higher titers. We tuned the safety profile, removing as many virulent genes as we could get our hands on in order to make the drug or the bug safer, excuse me. Purity-wise, we like to say pure in, pure out. If there's a pathway we've seen contributing to an impurity from the get-go, we removed or altered that pathway so that your product profile is good, and this is a mass spec profile of an unpurified lysate protein. So from the get-go, you have a high purity target. There aren't many post-translational modifications in E. coli, but there are some, and there are some other ways that you can start out with low product and process-related impurities. And we summarize this on the next slide as well. Overall, we see roughly log-fold improvements in titer. This varies by modality. If you're expressing solubly within the cell as an inclusion body or secreted, like you might do for peptides, we see big improvements compared to what we started with. And the more cells and the more per cell is what gets us to roughly 20 or more gram per liter titers intracellularly. And we'll specifically talk about some of the peptide titers we're able to get later on in this presentation. Pure coli is part of PUREplatform, your comprehensive solution to protein expression. PUREplatform, we like to say, is everything you need to make a protein. It comprises pure plasmids, pure media, and pure coli. And even within these three different elements, there is a library of sub-elements that we screen through to optimize your particular molecule of interest and create that bespoke solution for you. And we'll talk more about what that means specifically for peptides. We've got these libraries of different, not only standard genetic elements on plasmids, but libraries of tags and such that we'd apply to a peptide in order to optimize its expression. This table summarizes all the various attributes that we're proud of with respect to PUREplatform. Specifically, the growth rate or doubling rate is about 50% faster, which we estimate saves about half a day per batch, potentially more depending on the scales you're trying to go to and the number of generations you need to get through. Titer-wise, we've seen on average 5 to 20-fold improvements. This can vary up or down from there depending on the specific molecule, but certain cases we've seen up to 50 gram per liter. As I'll talk about, we tend to find that gram per liter titer is lower with smaller molecules. The peptide case study I'm going to talk about is 7 gram per liter expression titers. Product impurity-wise, as I mentioned, we've tuned many different pathways to reduce various product impurities that we've commonly seen so that you start off with a more pure product from the get-go. Process impurity-wise, we have 5 times fewer virulent genes or virulent-related genes. We don't require antibiotics. Our plasmid and host strain can pair through a complementation system. We find 5 times reduced antifoam is required with our bug, creating added robustness and reducing or possibly eliminating that process residual, which reduces testing and clearance needs. And we have, from the get-go, about a 3 times better DNA, HCP, and endotoxin ratio of product to these impurities compared to common E. coli. Stability-wise, we've removed many instability-related genes to stabilize the bug and ensure what you're getting from that single colony expanded to a research cell bank, master working cell bank, and whatever scale, 2, 20, 100,000 liters, that you get the results you expect without any sort of generational instabilities. Scalability, we've taken this and proven this to CGMP scale and shown the robustness of less foaming and aeration needs. If you have any questions, we've got case studies and all these different things. Please reach out to us and we'd be happy to dive in a little bit deeper in any one of these areas. Moving on to peptides, I'll talk first about the challenges with synthetic production and our opportunity in microbial fermentation. There's a growing peptide market dilemma that traditional peptide synthesis technology is not positioned to meet the growing market demand. The general thought is described by this graph here on the left, where as you grow in peptide length, adding more and more amino acids, the raw material cost, so just the cost of raw materials, not including service fees, grows exponentially. And the reason for this is even if you have, say, a 95% yield at each step of adding one amino acid after the other, 0.95 to the power of the number of amino acids, say 30, is a low number. And these protected amino acids can be quite costly. So as this grows exponentially, I'll show on the next slide, microbial fermentation does the opposite. Projecting into 2030, especially around the GLP-1s that have taken the world by storm, there's a projection of roughly 60,000 kegs of peptide material, so massive quantities of mass. Organic solvent waste is approaching 1 million kiloliters per year, which is an environmental hazard that we're going to have to address. And raw material cost, this totals about $50 billion per year for roughly a 30 to 35 amino acid peptide, which is what the GLP-1s commonly are. So any amount of improvement in this space could have huge ramifications financially as well as environmentally. And I want to highlight on this slide that scaling only is going to reduce the COGS from service fees. These are inherent environmental and material cost challenges that will persist at any scale and arguably grow, be more prevalent at larger scales. Conversely, fermentation raw material costs are inherently low. So whereas solid phase peptide synthesis, the raw material cost grows up, it tends to go down with fermentation, and the reason for this is that yields tend to be higher with longer amino acid chains. I've got this gray box here, which is roughly where the GLP-1s commonly are. If you take these raw material costs and you calculate the overall costs, the service fees plus the raw material costs, we estimate these costs at various scales from 2,000 to 20,000 and 100,000 liter fermentation. So you see by fermentation, the costs drop because most of the cost is driven by service fees, and this goes away as you have larger and larger scales, larger and larger economies of scale. But because the raw material costs are what are driving solid phase synthesis, they tend to be pretty similar at different scales. Now, traditionally, at lower fermentation volumes, it does not make sense, and we'll talk about some of the challenges on the next slide, to do fermentation. You can see the 2,000 liter orange bar being much larger than solid phase synthesis. However, when you have a cost-reducing engine like PUREcoli that can reduce the cost of goods by 5 to 10-fold, now all of a sudden it makes sense, even at the smaller scales and especially at the larger scales, to do microbial fermentation. So in conclusion, the raw materials themselves are simple salts and are cheap for fermentation. The bug is manufacturing all of the amino acids and putting them together for you, leveraging biology. But the feasibility of peptide production is driven by what yield you're going to get. And that's where we are excited to present this case study information for you in the coming slides. Some of the challenges, what's prevented people from doing this in the past is first, degradation. These peptides are small, they're unstructured. The bugs tend to see that as food. So they have proteases and such that chew it up and use it as nutrients. We have pre-built protease knockout strains in PUREcoli, and we've got further strain optimization going on all the time to tune specific strains and library of substrains that we can screen through for any given peptide to find that optimal system. And you can also optimize with protection tags. This isn't necessarily new. We have our own spin on it in flavor and library that we leverage. But really what differentiates us more than the tags is, again, the PUREcoli cost of goods engine that other people don't have, where we can get high biomass and strong expression from our bug, giving us that 5 times or so lower cost of goods. And that's addressed, shown here on the low titers. Next row, inherently high biomass stability and expression. Purity-wise, as well, that I mentioned, the inherently low impurity burden and secretion as well get you started off on a more pure foot, so to speak. Specifically, microbes like E. coli are traditionally bad at secreting, but we've proven that we can secrete a large peptide or small protein at about 8 gram per liter titers. And then we've got secretion tags layered on with protection tags that we'll talk about in a moment. So the aim of this work that we'll now get into is to demonstrate KBI's process for optimizing microbial peptide expression agnostically, and we're going to use Liraglutide as an example and provide a cost of goods estimate. All right, optimization of Liraglutide in pure coli. We're going to go through our expression and tag screening approach, scaling into an AMBER 250, give you a preliminary cost of goods analysis, and talk to you about our challenges and our future work lying ahead of us. So, we've developed a library of screening tags to optimize liraglutide expression. In this study, we screened about 8 different secretion signal peptides, 2 different affinity tags, 6 different charge tags, 12 different insolubility tags, if you will, or tags that direct to the inclusion body fraction or insoluble fraction, and about 20 different combination tags. So through combinations of these various tags, we can really design the purification. We can modulate the solubility of a particular peptide, as each will probably have their own biophysical properties. We can modulate and alter the purification scheme through multiple or single affinity tags, followed by charge tags, which lend for better chromatography on, say, a CEX if it's a positive tag. And altogether, I've got an example shown with this blue coil-looking structure representing the peptide of liraglutide with the tag. Roughly, we'll show on the next slide that the tag and the peptide are similar sizes. And the linkers that we looked at are cleavable either chemically or enzymatically. And so we are considering that. So it's the end-to-end solution that what you're going to end with after the purification is the native peptide. Tag optimization for liraglutide. I've got an example gel here where we screen through on this gel 5 different insolubility directing tags so that you can see the main product of interest is in the insoluble fraction. I've got a red arrow or orange arrow right where the product of interest is coming off. You can see a little bit difference in molecular weight, but these tags are all of similar molecular weight. There's some differences in expression profile, and this is our first round of optimization. We imagine that for any given peptide, we probably want to screen all 5 of these tags again to confirm which tag is the best. We don't envision that tag 2, although it won for this peptide, would win for every single peptide. At a high level, our major finding for liraglutide was, as expected, without a tag, you get minimal to no expression, and that we hypothesize is because the bug is chewing up the free peptide, as it has been reported to do in the literature and through others. With just a charge tag or just an affinity tag, we also see minimal expression. With a signal peptide, we see some expression to the extracellular space, but with inclusion body tags, as we'll call them, or the insolubility tag, we see these are the best at protecting, and that's why I showed this gel here to give an example. We also layered together combination tags of signal peptides, insolubility, and affinity, and believe that's our best for tuning protection, solubility, and downstream process. To optimize your overall cost of goods and hit your purity target by design. So the vision of a workflow for generic peptide optimization is that commonly when we're developing recombinant proteins, we're looking through these various host strains represented by those microbial cartoons and various genetic elements on plasmids, where you're looking at different promoters, origins, codon optimization strategies, we call them helper genes, etc. This is our common workflow at KBI Biopharma to do cell line development. And with peptide expression, we're doing that exact same thing, but multiplied against the different tags that we'd want to screen to optimize the expression of that peptide. Our different service offerings, you can reach out to us to learn more, but we've got 4 different service offerings that vary from 8 to 96 different plasmid strain combinations that can get you down to 4 to 12 or research cell banks that you could use for scaling and taking the next steps to get your peptide with its optimal tag, considering your holistic process. And the timeline for this is, depending on your scope, 8 strains is roughly a month and a half, up to 96 is closer to 3 months of development time. This was scaled into our standard platform Amber 250 fermentation on some of the lead candidates. Here's an example set of trends on temperature, pH, agitation, dissolved oxygen, and O2 full flow, showing rather minimal oxygen needs, roughly an OUR of 250, potentially even less. So rather conservative aeration requirements that should be amenable to roughly any scale. And we find works pretty robustly at all of our scales that we have, up to the 2,000 liter fermenter. And that there's opportunity to push this even further. Typically, we go closer to 300 for an OUR if we're really trying to optimize titers and push things further. But nonetheless, this gives us a starting point for optimizing biomass and expression. Here are the biomass curves for 2 different tags that we have here. Data-wise, we've got other tags that we've also expressed and that we're continuing to look at. Again, considering not just the overall expression from the fermenter, but the overall process through downstream purification. But you can see the biomass getting up to an OD600, nearly 160. For pure coli, at cGMP scale, we've seen biomasses of OD600 of 300. So just by biomass, we feel there's an opportunity to optimize titers nearly twofold. But this, again, is a good starting place for initial expression studies. For the different tags, both insolubility tags, but of different sizes, you can see in the insoluble fraction, the peptide plus tag band of interest coming off, marked in purple, and against the whole cells as well. Liraglutide, for reference, is also shown. Again, it runs lower because it does not have the tag as part of the construct. Next, we applied what we like to call a midstream screening to find the optimal preparation of this insoluble fraction. And we envision this would also be our agnostic way to optimize the overall process for a peptide directed to the insoluble fraction. At its heart, what we're doing is screening various pHs, detergents, conductivity, denaturants, and combinations. And what we're able to do here is generate a process where we optimize the wash to remove as many of the impurities as possible, and then end with a process that's able to solubilize that insoluble fraction without affecting the capture column negatively. Our findings were we found some optimal ranges for both high and low pHs of solubilizing HCPs and separately solubilizing the tagged peptide. We found certain detergents that can solubilize HCPs, but not the tagged peptide, but the right pH detergent combination, which can solubilize our tagged peptide. Conductivity in this case drove the tagged peptide insoluble at any condition, which we could leverage to our advantage to try and keep it insoluble while we're washing away impurities and wash away DNA and other things that need some charge shielding driven by conductivity, especially guanidine hydrochloride, for example, which imparts conductivity and can solubilize HCPs but not the tagged peptide well. And then finally, we ended with, as I mentioned, a certain combination of pH and detergent and denaturant that gently solubilized the peptide so that we could load onto column 1, and that data is pending. Overall, here's some high-level numbers. From the fermentation stage, we got about 7 grams per liter titers. We got about a 90% yield through the harvest and IB washing stage. 90% yield, which is common for IB processes, we find, especially as we designed the washing to maximize yield. We have a 2-column process that we're planning, which I've got asterisks here. And that, as I said, that data is pending. It's affinity based, and we're using our experience there to estimate that through 2 columns and with the starting purities that we've already got, we should have this yield or better. But using that preliminary estimate, as well as the 7 gram per liter, I should say that that 7 has an asterisk there because that's the titer of the peptide with the tag on it. If you were to remove the tag, the titer would be roughly half of that, 3.5 gram per liter. And so the cost per gram of purified peptide is the estimated cost per gram after you've cleaved off the tag of just the peptide that's been purified. This includes service fees as well as all raw material costs for resins, for media, etc. At the 2,000 liter scale, we estimate about $700 per gram, and if you were to scale up from there, you could get down to $200 and then $100 per gram, which we feel is a great initial go at this. As mentioned a couple slides ago, we think we can possibly improve about 2x on this potentially by increasing fermentation titers and tweaking things to, optimize yield. But nonetheless, a great starting spot. We've done some mass spec to show the correct product as well. And we're just excited to finish the chromatography and verify through the purification process and assess the purity profile. In conclusion, we see a great opportunity in microbial fermentation for producing peptides in a much greener and more cost-effective way than solid-phase peptide synthesis. Peptides can be stabilized and purified with the appropriate tags, and we believe we've a system where we have a library of tags and strains that we can screen on any given peptide. We applied this concept to liraglutide specifically, but any given peptide will have a different length. It'll have a different stability or resistance to degradation from the cell. It'll have a different solubility. And so this process of screening to arrive at the optimal solution we think is applicable, agnostic of peptide. The lead candidate shows about 7 gram per liter titers of tagged liraglutide, and the washed IB rather clean by SDS page. The PUREplatform COG engine is what drives this all. It's been proven before on small proteins and large peptides, and we have this case study now showing its applicability to peptides of about 30 amino acids, and that we can get to 5 times lower cost of goods than common E. coli. Our future work is to confirm these yields, especially in the purification and the quality through purification, and then scale this to our pilot scale for cost of goods confirmation. Pardon me. Thank you very much for your time. Once again, if you would like to take out your phone and scan the QR code here, there's a short survey that would help us better serve you and connect with you more. Thank you so much for your attention. I'm happy to field any questions now.